Tiny brain cells, massive impact. Stress-sensitive neurons control whole brain function. A newly discovered population of nerve cells, called type I NNOS neurons, appears to exert a powerful, system-wide influence on brain activity and blood flow. Research suggests these cells are critical for maintaining healthy brain function, with potential implications for understanding sleep disorders, neurodegenerative diseases, and the effects of chronic stress. The discovery: a small group with outsized influence researchers at Pennsylvania State University found that these sparsely distributed neurons, primarily located in deep cortical layers, play a surprising role in regulating the brain's spontaneous oscillations, the rhythmic pulsing of blood flow that delivers oxygen and clears waste. To understand their function, scientists selectively removed type I NNOS neurons from the brains of mice and observed the effects. What happens when these cells disappear? The results were striking. Without these neurons, mice exhibited significantly reduced blood flow, weaker pulsations in blood vessels, a process called vasomotion, and decreased overall brain activity. Slow brain waves associated with deep sleep delta waves also weakened, and communication between the left and right hemispheres became less synchronized. In your brain, arteries, veins, and capillaries help move fluid around by constantly dilating and constricting every few seconds, which we call spontaneous oscillation, explains biomedical engineer Patrick Drew. After targeting and eliminating a subset of these neurons, we observed a significant reduction in the amplitude of these oscillations. Why this matters? Linking brain health to key processes, the study suggests type I NNOS neurons are crucial for several fundamental brain functions. Sleep. Delta waves, which are weakened when these neurons are removed, are closely linked to deep, restorative sleep. Waste clearance. Vasomotion is vital for flushing metabolic waste from the brain. Impaired vasomotion is a hallmark of neurodegenerative diseases like Alzheimer's. Brain synchronization. Healthy communication between brain hemispheres depends on these neurons' activity. The researchers conclude that the loss of these neurons could contribute to the development of sleep disturbances and neurodegenerative conditions. The stress connection. A potential environmental trigger, type I NNOS neurons are particularly vulnerable to mental stress. This raises the possibility that chronic stress could lead to the loss of these cells, contributing to poor brain health over time. Reduced blood flow is one of many contributing factors to reduced brain function and neurodegenerative diseases, Drew says. While we know aging plays a major role in this, losing these rare neurons to chronic stress could be an unexplored environmental cause for poor brain health. What's next? While these findings come from mouse studies, the researchers believe human brains may operate similarly. Future studies are needed to confirm this link and explore whether disruptions to type I NNOS neuron function play a role in neurological disorders. This research highlights the critical importance of maintaining healthy blood flow and protecting these tiny, yet powerful, brain cells from the damaging effects of chronic stress.